Parsa.
Good to see you again.
We do these on Friday afternoons, and for those of you that don't know, Parsa and I love talking to each other. So it's like the last thing we do
All
on
the
Fridays.
time, only on Fridays.
And so I'm always in a good mood. He's always in a good mood. Today, we're going to talk about clinical trial matching. And Parsa, I want to start the conversation out by saying, why are we talking about this? What is such a very specific thing got to do with the learning health system, and how does one help the other?
Yeah, I think the broader context for this is patient screening and finding the right patients for the right studies. And that's
So
why,
when we're talking about clinical trial matching, we're not talking about the clinical trial management, and we're not talking about recruiting patients. We're
no.
only talking about identifying patients.
Yeah, it is about, I mean, the process of knowledge generation, evidence generation in learning health system is all about being able to answer questions about patients. It is critical to find the right patients for the right questions and make sure that you're studying the group of patients that you can generalize across either problems that you're trying to solve. And that requires these kinds of deep understanding of people and matching them to kind of questions and problems that are being solved, or trials, if you will, in the traditional sense.
So you've been tasked with solving this at Montefiore. I just always sort of thought that, clinical trials and clinical trial recruitment and this kind of thing, that there's, it's been going on for a long time. There are dedicated companies set up to it. I would always have thought that there was already software out there to sort of do everything you need to do here. You have found that is not the case?
I think just the fact that there are many, many people trying to do it tells you that the problem is complex and still not solved. Am I claiming that we have solved it? I don't know, but I think that the answer is maybe. Because what we have done is to build a system that really understands the problem in a very deep way and tries to bring all of that deep understanding and deep knowledge of the patients, the research, the trials, the matching, building trust, all of those together to execute on that.
So let's dive a little deeper on that. If I understand this correctly, what you're saying is because you have built, or you are building, or you're creating a learning health system at Montefiore, you're saying that your ability to recruit patients for clinical trials using the tech stack that you've built is improved.
What we have built is a generalizable and scalable way of solving the problem that can be expanded, extended, and over time really reduce the problem to something that can be not only solved for clinical trials that we started talking about, but also any patient matching problem in healthcare system that requires identifying the right set of patients. What we have done is to create a system that can change its frame of mind as you change your frame of mind. But of course, the very first application, the most important application that we have started is around matching the clinical trials, starting with oncology trials, starting with breast and lung cancer trials. So,
I'm going to stop you there, because what I like about this is that what you're saying is you've got a generalizable solution, but we're going to look at a specific example of it to illustrate the generalizable principle, which I love.
Exactly.
So let's start with what you've built. And I took a couple of notes, so excuse me for looking down here. And the first note I have here about this product is the understanding trial inclusion-exclusion criteria requires complex and nuanced clinical and domain-specific knowledge.
Yes, but maybe before that we can actually say what its name is.
Yeah, the product has a name. Called it?
Yeah,
you
the
called it
technology
something?
has a name. Yeah, we call it oTESSA. oTESSA is O plus TESSA. O for oncology, and TESSA for trial evaluation and smart screening algorithm. And I separated them because what we have built is the TESSA. It's just trial evaluation and smart screening algorithm, but we are starting with oncology trials first, therefore, hence oTESSA.
So you want to identify patients based on clinical trial requirements. Those are all up on clinicaltrials.gov. All of the inclusion and exclusion criteria. What's the
Yeah.
problem here?
Well, that's the easiest thing that comes to mind to do, right? There's a set of descriptions on what the trial is about.
Yep.
It is explained in the document that you can easily access through trials.gov API. And it can tell you all about what you need to find in patients that you're interested in. And that's also the very first problem for any smart algorithm, because those documentations are human readable. They are designed to be understood by oncologists, research coordinators, and practitioners in the field of oncology. And even more specific than that, in a breast trial or in a long trial, it is written to be understood by oncologists that do only breast or do only lung cancer. Very highly specialized language. You need to know every specific medical terminology. You need to know how to interpret it. And you need to know where in clinical documents and EHR system and in different modalities of information that you have inside an EMR, you can actually find those information.
Have you got an example of something you can show me?
Yes, you can look into probably a breast cancer trial criterion. I sent you an example earlier. We can actually review it together if you
Oh,
can.
hang on a second. Yeah, let me pull it up. This is one criterion from a breast cancer trial, right? It's
Yes.
HER2 positive status will be based on pre-treatment biopsy material and defined according to the current American Society of Clinical Oncology, ASCO, College of American Pathologists, CAP, guidelines. Hormone receptor positive status can be determined by either known positive estrogen receptor, ER, or known positive progesterone receptor, PR status. Hormone receptor negative status must be determined by both negative ER and known negative PR.
That's just one criterion. Most clinical trials have at least 10, average 10 clinical trials. This one that I actually grabbed it from has 37 criteria like
But
this.
am I naive in saying that's what large language models were built to do? Really?
Yeah, but in order to be able to understand this criterion, you need to have a lot of domain knowledge as well as cast a lot of clinical judgment to understand exactly in your institution, in your setting, with your environment and patients and so forth, what this criterion means. Where
Sure.
are you going to look to find this information? And of course, you need to also understand those criteria based on those guidelines that were named in that criterion. A lot of cognitive capacity is involved to identify only, to understand only one of those criterion. And a typical clinical trial has, in average, somewhere between 8 to 10 of these. This one that you actually read was from a trial with 37 criteria like that. It is cognitively loaded. It's very hard for anyone to understand the full context of a single trial with 37 criteria and then find the right
Okay. Okay. So
match
I
for that.
understand why that's very hard for a person. So how did you solve that with technology?
What we did was actually to create an agentic design, meaning multiple smaller agents, each one with a really good and specialized task that they were trained and refined to do, to look into a very complex criterion and break it down into smaller pieces, identify what that piece involves. Sometimes criterion is about cancer type, sometimes it's about cancer stage, sometimes it's about cancer stage and the tumor size, sometimes it's cancer stage as well as the history of therapeutic modalities that the patient was on, and so forth. It breaks it apart, identifies what are those parts, and assigns an agent that is specialized to that part to interpret it and create a logical relationship understood by the machine and structured in a way that can be pushed to the next level, a next, let's say, team of agents that could take these instructions, put together, and execute on them. So you basically use a army of specialized agents to take that human language representation of what the clinical trial is, add domain knowledge, add technical knowledge, add interpretations that are specific to your institution because of the training that they know, what kind of documents do you have, what each one of those documents entail, and so forth, and create a new representation of the criterion that is built for machines, and then pass it to the next level. That constitutes of our understanding of what are we looking for.
Okay, we've got it to the stage where our little AI ensemble has understood the criterion and got it into a position where it is machine understandable. Now what?
The next question is, where do we find that information? No matter how we are going to find it, right? But where do we go to find it? Is that the question, right? In healthcare, we create hundreds of
And when you say where do we go, you mean like where in
data
the
sets, data
EHR? Is
systems.
it structured data or unstructured data?
Yeah,
What notes? That's what you mean by where?
exactly. What are the data sources that are in the best position to answer these kinds of questions?
Yep.
In healthcare system, just in Montefiore, we create north of 150 different type of clinical notes. And every single one of them may have a snippet of information about how this patient may or may not match. Patient may have all of the inclusion criteria in one, but for exclusion criteria, you may have to survey a massive amount of information to see if patient doesn't have allergy to something, because that information may be embedded anywhere, quite honestly. And then there is massive amount of structured information inside EMR, that from biomarkers to diagnoses to other findings, observations, devices. So there are really large data sets that can be everywhere that you can harvest for this task. In order to solve the problem, and it is not like intuitively one may think that the more information you have, the better matching. If we just bring everything together, you will have a better chance to answer these kinds of questions, but empirically, that's not true, especially for two reasons. Information changes over time. I may not have allergy, or my allergy may not be found yesterday, but now I have an allergy. And if you bring both of these documents together, you're going to have two documents that says you have an allergy and the other one says you don't have allergy. So that temporality changes the nature of the information in a way that you have to have a temporal idea about where to go. In clinical notes, we also have this other problem, which is people copy paste, or when they are documenting care, they don't know about the latest information about the patient. So they create documentation that is not on time or timely, which creates inconsistencies. You have an earlier document that says you are in stage 2B, and then you have a later document, the newest document, that says patient is stage 2. They copy and paste information from different places without paying attention to how accurate and how recent these information are. And they're now all replicated redundantly everywhere. And now you're bringing every one of them. Which one of those versions of information are you going to pick? We have identified the problem of information overload, inconsistencies, disaggregation of information, that the same tumor information can be found in 20 different sources. It might be in pathology and radiology and clinical notes and follow-up notes of the patient. Which one is the most authentic, authorized source of information for that one data point? So
And
what I'm trying to say,
what's interesting about that is you use the phrase information overload, which is used with humans, but you're using that now with machines, which is interesting.
of course,
Yeah.
for all, I think, purposes, we are talking about human level intelligence necessary for solving these problems, and
Yeah.
whatever can impact humans will definitely also impact here, including the biases, errors, everything. We have created a, let's say, patient context mechanism, which is where you go for information rather than just go everywhere. And that patient context has a vectorized version of all of the clinical notes that we think have the highest information value for solving these kinds of matching and screening problems, rather than everything that may have some information, and indexed those notes temporally in a way that they can be reviewed, let's say, descendantly, in terms of temporal indexing. We have also created a feature store that combines with this vector store that allows us to extract essential features that are critical for oncology, like tumor type, tumor stage, therapeutical modalities, with specific timestamps, all of the important biomarkers such as HER2 and so forth status, in a way that those features can be queried, and all of the texts and explanations of those features that might be necessary can be queried at the same time through this patient context.
So you segmented the data so it can be machine readable. You've got these machines to understand it, read it. Now you have to match up the patients. Talk to us
Yes.
about that.
So the matching problem is the step after information retrieval, right? So what we did so far was we understood what to look for. We understood where to go
Yep.
to find what we are looking for. And then you have to go and find pieces of information as per criterion that you are looking for from the patient context that can be used to answer the matching problem. You are not still
Yep.
matching. You have agents that know exactly what they need to do for the parts of the problem they need to solve. They have no idea how the other agents will use that information. They're just very good at what they're trained to do. In this case, given the criteria, knowing where to go to find the information exactly as it is required as per criterion. It doesn't really know what the logic is and how to match, but it brings all of the information together and passes to the next level of agents that can do a comparison between what we needed and what we got from this process.
That's kind of incredible. You built all of these different agents doing all of these different micro jobs that are, building on one another. Why is it necessary to have all of this structure and all of these little pieces that it's almost like you're building a jigsaw puzzle?
Yes, there are many reasons. One is that it is a really complex problem to solve, and expecting a model, even with 70 billion model or hundreds of billions of models to solve this problem can be computationally very intense. and sometimes even not possible. It's just a lot of iterations and a lot of validations that needs to happen.
And I guess when you
Secondly,
say computationally intense, a business person would hear expensive.
expensive, yes, and you're gonna melt glaciers to match a patient with the
Climate
amount of
damaging and intensive. Yeah, okay.
exactly. So you don't want to do that. Second reason is that you want to make sure that you have mechanisms built into your system to eliminate potentials for hallucination, right? You want to make sure every single bit of information that is being considered by your large language model is factual and not made up. So we build those traceability into this process itself to be able to eliminate potentials for bias. Third reason why you would do these kinds of design, other than just increasing the accuracy and performance metrics, is ability to justify and provide explanation. So by the time that you get to the end of the pipeline, you have a documentation provided by the algorithm itself of exactly what was the workflow that was used to find information, package information, interpret the information, synthesize the information, do the matchmaking, down to the validation. You get a report on every step created by the algorithm itself.
Wow. So you're in a way, you're really opening up the black box. I mean, so often these large language models, you just put it in a question and you're going to count an answer. It's just, I don't know why it said that. But in a way, because you've broken it down, you're giving transparency into what
Yeah,
needs to.
we incorporate a flashlight into every step of
That's
this
fantastic.
process walked by language models. So there's nothing in the dark that is happening without us knowing. So that's just one step of the matchmaking. And we haven't still done matchmaking. What we have done is
Let's get to matchmaking.
we have found information, we have validated information, and justified that this information is actually good information, useful information to do the matchmaking. And you pass that to the next level, which is now your judge. You have a judgment system that its job is only to do the match. The judge also determines whether or not it's going to.
And to be clear, this judge is now armed on the one hand from this pipeline that's come in from the trial and it's disaggregated that. And on the other hand, this pipeline that's come in from the patients where it's gone and taken all of the conflicts out and it's written it down in a way that it can understand. So you have two clean data sets that the judge is looking at. Is that correct?
Exactly. The agentic system that I was describing actually connects these two pieces together
Yeah.
before giving it to the judge. So it basically matches information that is retrieved from the patient context to the criterion or sub-criterion that requires that information, and says, you need to make a decision about this, and these are the facts that I have to be used for this criterion, and justifications. This process binds these two pieces of information together in a way that not only everything is connected, but also justified and explained. And when we pass it to the judge actually has three sets of information: what was asked, what was found, and the justification why those things that are found are good enough for a decision making. And then the judge is good enough to be able to basically understand the logical structure of the criterion, map that to the data that we have found, and determine whether or not sufficient evidence have been found to be able to do the match. And the judge basically says, yes, this is a match, or no, this is not a match, and justifies its own act. It creates a justification for its own decision making. we don't still trust it, right? Is okay, thank you very much. Let's see if a second validator, the examiner, will agree with you.
Oh, no kidding. And then does the examiner give like a percentage that he's sure that they're right? Is that how that works? Or how?
No, examiner actually justifies the judge and says, is the justification of the judge is supported by the evidence provided to the judge.
So then what happens? So then does it just spit out the answer to the clinician? What happens at the next step?
Yeah, well, no spitting.
No spitting. Fair enough.
We put all of those information in this registry of all of the trials and patients that we have attempted to match, and we invite domain experts, clinicians, research coordinators to review all of the matches between the patient and the criterion, and basically say, do we accept or do we not accept this matchmaking by the judge? Not only they can accept and reject the matches, they also provide their input about two other things. One is you want to make sure that your justifications are sound and acceptable and really up to the point. You rate that justification, as well as I mentioned, we bring forward all of the evidence that we've used, all of the data points that we've used to cast the judgment and inference as part of the judgment and inference. We show all of those evidence to our users and say, okay, well, we said the patient is matched, but this was exactly the kind of evidence we used. So you create an ensemble of all of the data that you have used to basically match the patient. And we ask our reviewers to say whether or not these are acceptable data and support this judgment. And they rate that. The reason for why we do this is we have the reinforcement learning with human feedback in place that allows to cycle back all of these feedback to the model training and make sure that over time the system can align itself with the expectations of the human judges and human reviewers and gets better and better. The second reason for that is based on different environments, different cultures, different institutions, there might be differences of opinion about what is acceptable, what is not acceptable, what is a sound judgment, what is not, what's the level of data that you need to be able to justify matching a patient and so forth. And you want to provide a way of bringing all of those institutional and localized context and introduce that to the model training as well, which is part of the process.
When you look at all of these criterion, I think at the beginning you said the one that I read was one of 37, something like that.
Yeah, it was a trial with 37.
Do they really have to match every single one of those criteria? Are all the criterions created equal?
Very good question, actually, because the answer is no. Not all criterion are equal, and that's where actually human judgment, clinician judgment, is essential for matching and absent from almost all of the traditional technologies. Let me give you an example. In a breast cancer trial, you definitely need to be a female and should have breast cancer to match, right? It's just hard fact. You can't escape that. So
no.
Obviously, those are hard criteria that should be matched. If their clinical trial requires to have an MRI scan of certain kind prior to inclusion and you don't have that MRI, should we disqualify you? No.
Oh, I can think of a whole bunch more. If you have high cholesterol, you could go on cholesterol-lowering measures.
Exactly. You have high creatinine, you can be hydrated and tested again. So there are a lot of soft criteria that you may not match now, but with a little bit of an adjustment and coordination with the patient, you can actually overcome that. So these are things that we call soft criteria. One aspect of how we do this with oTESSA is that oTESSA, when it considers understanding the criterion and structuring it in a way that it can actually find information, it also assigns a soft or hard criterion justification and say, okay, don't worry, you don't have to meet this criteria. If you didn't meet it, these are the three things that we think we can do to still qualify for you, and provide justification, provides suggestions on what to do if patients didn't match this criteria. So when we do all of this, after all is done, we create a percentage of criterions that the patients have matched based on the hard criteria. And we also provide percentage of criteria that the patients have not matched based on the soft criteria, with suggestions on what to do to prepare the patients, maybe 2 months down the road, maybe after another procedure, to be recruited. So you're not losing patients to these little things. You are basically creating an environment in which you can cultivate a pool of applicants that you know what to do for each one of them to benefit from the clinical trials that you have in the institution.
Last question: how's the rollout going? How's it being received? How, as a product, is it up and running? How's that going?
Yes, we have trained oTESSA so far between breast and lung cancer trials. More than 40 trials are being essentially reviewed every day. You can imagine when you have hundreds of patients and hundreds of trials, you have tens of thousands of matches to be made. So it's again, still we are melting some glaciers. So it's a massive operation. The way we do it is every week before those clinics open, we know who are people that have appointments in those clinics, and we match them for trials that those clinics are supporting. So it is matching the right patients to the right trials. It's not all patients, all trials in hopes of finding some match. And we have started integration to Epic, our EHR system. So when a physician sees a patient, the patient is in front of me, I want to know if we have any open trials that I can offer to this patient that patient may benefit from. So that just-in-time matching is also going to be possible at the point of care. We're collaborating with our excellent lung cancer team to do this, and we are very excited about it. And the last but not least part of our development is to do this in real time for patients that are in ICU. For example, for acute care, you only have half an hour to enroll patient in this line of therapy, to give them this new drug that may be beneficial for that patient right there, right then, using fast pace streaming data that's coming through. And that we are very excited working with our critical care team to be able to do these kinds of just-in-time matching in real time as well.
It's fantastic. What I love most about this, although I love the product, is it's such a clear manifestation of a learning health system, that you can get all of this data about a patient in real time, and you can have, demonstrable, understandable products that come out of it. And I love that.
And continuously improve it.
Yes.
Continuous cycle of improvement is built into everything that we do here, in terms of just the architectural understanding of what tools are good for learning health system and what are the components and functionalities that will align with the ethos of learning health system.
Parsa, it's always a pleasure. Enjoy the rest of your Friday. I love our little chats. I wonder what we'll talk about next week.
Yes.
Yeah. Okay.